Welcome back, Bio30 Prep. We're going to continue our discussion of the circulatory system and immunity. And in this lecture, we're going to be looking at the function of blood, or the composition of blood. In your textbook, it's pages 282 to 291. By the end of this lecture, you'll be able to describe the composition of blood. You'll be able to identify the role of various constituencies of blood. So you'll talk about the function of plasma and of the blood cells. You'll describe the role of the circulatory system in maintaining homeostasis. When we look at the components of blood, about 55% of your blood is plasma. 90% of that is water, and 10% of that are blood proteins, glucose, vitamins, minerals, dissolved gases, and metabolic waste. So all of those molecules that are responsible for either helping cells do their job or the waste material from after they're done their job. So important blood plasma proteins that we're going to be talking about are albumins, globulins, and fibrinogen. Albumins play an important role in osmotic balance, or water balance. Globulins are involved in antibodies and immunity, and fibrinogen is the blood clotting protein. The other 45% of your blood is cells. 99% of that are erythrocytes, or the red blood cells. Then there's also leukocytes and platelets. So here's a breakdown of what blood looks like if you were to spin it in a centrifuge or separate out the components and put it into layers. You get about 44% of your blood is red blood cells, about 1% white blood cells and platelets, and then 55% would be the plasma. Here's a breakdown or a table showing where cells are made, or where blood cells are made, their basic job, and the kind of numbers you'd expect per cubic millimeter. So this is quite a small volume. If we take a look, red blood cells made in the bone marrow, and if we take a look at all of these blood cells, you're going to see that bone marrow is common to all of them. So all blood cells are made in bone marrow. Some of them are made in other places as well. Lymphocytes are made in the thymus, and platelets are generated in the lungs as well. But if you remember that red bone marrow part, that's the important part. All blood cells can be made in the red bone marrow. In terms of numbers, we just want to talk about the number of red blood cells. The average male has 5.5 million red blood cells in a cubic milliliter, as compared to 6,000, 2,000, and 250,000 platelets in the same volume. Size-wise, the largest are the white blood cells, granulocytes and monocytes being the largest, lymphocytes following a close second. The smallest blood cells are the platelets. And then general function, red blood cells are your oxygen-carrying cells. White blood cells are involved in immunity. They're either going to engulf foreign particles or play a role in antibodies or defense function. Platelets, their sole function is clotting of blood. They live different lengths as well. Red blood cells live about 120 days before they're replaced. Granulocytes and monocytes, a few hours to a few days, depending on when they get used. Lymphocytes, it's not really known. The memory cells, and we'll talk about those soon, can stick around for quite a while, so it's not really known how long these live for, because our immunities can be carried throughout our life. Platelets last about 2 to 8 days. So for red blood cells, their primary function is to transport oxygen. They are the cells that contain hemoglobin. That red pigment in blood is from the iron that's present in hemoglobin, and hemoglobin is a protein. Without hemoglobin, only about 3 milliliters of oxygen could be dissolved in 1 liter of blood. So in the 5 liters of blood that travel throughout our body, we'd get about 15 milliliters of oxygen, and that would be enough to keep us alive for about 4.5 seconds, which isn't very long. No holding your breath. 1 liter of blood with hemoglobin molecules can now dissolve 200 milliliters of dissolved oxygen. That's a huge difference between 3 mils and 200 mils. So hemoglobin, extremely important protein in our blood, that's enough oxygen to keep you alive for 5 minutes. Once all the oxygen in your body is used up, cell death starts to occur. Without oxygen, there's no energy. Even though you might have glucose in your blood, oxygen, you have to have oxygen there to metabolize it, so no oxygen, can't get access to the energy, and cells start to die off. In terms of the structure of a erythrocyte, here's a picture of a red blood cell. What you'll notice is they're a biconcave disc, where they have this dip in the middle on both sides. What that does is it increases surface area, and if you look at it in kind of a 2D image, the two choices, I guess, would be a flat surface or a curved surface. And if you take a look, this here is a longer line. It has more area on it, so it increases the rate at which gas exchange can occur. More oxygen can diffuse into those red blood cells attached to hemoglobin, and when it's time to get rid of that oxygen to our body cells, it's easier to do that as well. So it increases surface area for increased gas exchange, internal and external respiration. They're enucleated, so they have no nucleus, and that provides more room for hemoglobin, so they're extremely specialized. I-Z-E-D. There we go. Extremely specialized. So the question is, how do red blood cells reproduce? If they don't have a nucleus, they don't have DNA, and if you don't have DNA, there's no mitosis. No cell reproduction. 280 million hemoglobin molecules can be found in every single one of those erythrocytes, and so if you're looking at the average male, 5.5 million cells times 280 million hemoglobins, that's in 1 cubic millimeter of blood. And we have 5 liters, which is actually 5. We'll just go with 5 liters. So, in other words, we have a lot of hemoglobin. Hemoglobin consists of two structures, a heme structure and a globulin structure. The heme is the iron-containing pigment. The globulin is the protein structure. Each hemoglobin has 4 iron molecules, which means each iron molecule combined 4 oxygens. So now we can multiply that by 4, and that would tell you how many oxygen molecules our body can carry at any one time, which is a really big number. The removal of oxygen molecules from the hemoglobin changes its shape, and then it changes the light that it reflects, and that's why venous blood, or blood in our veins, has a bluish tinge to it. Leukocytes, white blood cells, they're nucleated, so they have a nucleus, so they can reproduce. They're part of the body's immune system, and so their numbers increase when our body is fighting an infection. There's two major types, phagocytes and lymphocytes. Phagocytes, their primary role is to engulf foreign invaders. Their job is to find foreign cells and eat them, basically. Once the microbe is engulfed, digestive enzymes are released, and those enzymes destroy both the leukocyte and the invading cell. So these phagocytes are kind of like suicide bombers. The result is pus. So granulocytes, and there's 3 types, neutrophiles, basophiles, and eosinophiles. That's one type of phagocyte. The other type are monocytes. They can leave blood vessels and become macrophages, and these are important because macrophages, these stimulate lymphocytes. There's 2 L words, and they're both related to white blood cells. There's leukocytes, which is all your white blood cells, and then there's lymphocytes, which are specialized white blood cells. So, the leukocytes squeeze through a capillary membrane. It's called diapedesis when they do so, and they can move towards foreign microbes. The lymphocytes are involved in an antibody response against foreign invaders. And as we go on and talk more about immunity, we'll find out more about exactly what a lymphocyte is. There's platelets. They are enucleated, so again, they have no nucleus. Enucleated, this means without a nucleus. They're produced in bone marrow from cells called megakaryocytes, and they're fragile. When they rupture from striking a sharp edge, they start to activate, and they go through there's a long chain of enzymatic reactions, and we're going to look at a few of them. So they initiate blood clotting, and there's trillions of platelets moving through your body's blood vessels at any one time. When it comes to blood clotting, it can be looked at as being a homeostasis response, maintaining blood volume in your body. So their job is to reinforce weakened blood vessels. If a blood vessel gets cut or bruised or opened up, this is going to seal it off. So blood clots form when platelets rupture from hitting a rough surface, causing a series of enzyme-catalyzed reactions. The first one that we're going to talk about is a release of thromboplastin. Thromboplastin, in the presence of calcium ions and in your blood, there's already a bunch of calcium just sitting there doing nothing. It activates the prothrombin protein. It's present in the blood, produced by the liver, and the active form of prothrombin is thrombin. So what we're trying to do is a bunch of activation steps to get a blood clot to form. One of the reasons it's so complicated is that if blood clots were easily formed, they would pose more of a risk than a help. If blood clots form easily, then they might start forming when we don't want them to. So there's a series of steps involved in forming a blood clot. That thrombin cuts the fibrinogen, forming fibrin threads. So fibrinogen, it's already in your blood, it's sitting there, it's doing nothing, it's a protein. In the presence of thrombin, it's turned into fibrin threads. These form a net over the damaged area. Red blood cells can't get through. The white blood cells can crawl through the net in order to attack foreign invaders. Foreign invaders are generally kept out, and the fibrinogen is produced by the liver. So here's another picture of blood clotting. So if we just look at the steps, the first step that we would look at is a platelet impacts a rough surface. It's going to break open the platelet, and proteins are released. One of those proteins is prothrombin. So the protein we're talking about here is prothrombin. Oh, sorry, I lied. Well, I didn't lie. Here, I made a mistake, so we'll erase that line. And we'll go back. Here we go. Okay, so thromboplastin is released. Thromboplastin and calcium together convert prothrombin, which is a protein in the blood, into thrombin. And then thrombin does its thing. Thrombin and the calcium ions in your blood activate another protein. So it's a multi-step process. These aren't even all the steps, they're just a summary of the steps. This is what produces the fibrin thread, and the fibrin thread forms that tangled net which causes a clot to form. The clot dries, forming a scab, and then those blood vessels can heal underneath the scab, and once they're done, the scab falls off. When a blood clot does go bad, it's called a thrombus, and this is where a blood clot seals off a blood vessel. So this usually happens when an embolus or if a blood clot starts floating through the circulatory system, it gets into narrower and narrower arteries until it plugs an arteriole, and this causes localized cell tissue death due to oxygen depletion. And they're named based on where they happen. So a cerebral thrombus can cause a stroke, coronary thrombus can cause a heart attack. An embolus is the dislodged blood clot, and these are the dangerous ones. They travel through the circulatory system, and if they start in a large enough artery, as they travel down towards a capillary bed, they enter smaller and smaller vessels until they hit one that they can't pass through. And so you can have a cerebral embolus, coronary embolus, pulmonary embolus. The name just tells you where the embolus lodged inside the body. And this, of course, is going to cause damage to either the brain, the heart, or the lungs. The other part of blood is plasma. It's the fluid portion of blood, and it acts as a transport medium for blood cells, blood proteins, and nutrients. The blood proteins, and we talked about them a little bit, the albumins maintain osmotic balance, and this we talked about, this gets the water back into capillaries from the extracellular fluid. So that's one of the roles of albumins. Immunity is controlled by antibodies, which are proteins. There's also complementary proteins as well. Blood clotting is fibrinogen. These proteins are in the blood all the time. They get activated by usually other proteins or substances when something happens. It's also a transport medium for nutrients, such as dissolved sugars and amino acids. It contains the waste material from cells, such as carbon dioxide, as bicarbonate ions. And it interacts with a variety of other organ systems. Hormone transport from the alimentary canal to other accessory organs, from the brain to other endocrine glands, so our thyroid gland, our adrenal gland, and other glands are controlled by our brain, and what it does is it actually leaks hormones into our circulatory system. Those hormones then travel throughout our body and trigger other glands to do things. For the amount of dissolved oxygen in our blood, I've already said that we need to have oxygen in our blood, otherwise we start getting tissue death. Individuals can have between 4 to 8 million red blood cells per milliliter in their blood, depending on the gender and their environment. So the body actually monitors the red blood cell level by monitoring oxygen levels in the body. So this isn't going to cause us to inhale or exhale, but what this does is it monitors what our average oxygen level is and determines if that's okay. So the oxygen levels are monitored by the kidney. If there's low oxygen levels in our body, it causes the kidney to produce renal erythropoietic factor, or REF, so renal erythropoietic factor. It combines with the globulins that are already present in our blood to form a hormone called erythropoietin. Erythropoietin stimulates erythropoiesis, or the generation of red blood cells in bone marrow. So generate red blood cells, or erythrocytes. Things that can cause this: exercise, and this isn't necessarily just the exercise of running once, but people who exercise often are going to have more red blood cells in their circulatory system because they spend more time in a kind of an oxygen depleted state. If you're working hard, you're using oxygen more quickly, and your body gets used to that routine and produces more red blood cells. Living in high altitudes because the air is thin. High altitudes, same as thin air, there's not as much oxygen at high altitudes, so you need more red blood cells to make sure you capture as many as possible. Hemorrhaging, obviously if you lose blood, it needs to be replaced. So all of these can stimulate erythropoiesis. Erythropoietin can be administered externally, and this is one way to do blood doping. Instead of injecting blood, if you inject erythropoietic factor or erythropoietin, it does the same thing. It stimulates that red blood cell generation, and thereby increasing your red blood cell count. The more oxygen you have in your blood, the longer you can do strenuous activity. So here's an example, for example, of a homeostatic effect. If you're living at a high altitude where the air is thin, this stimulus is going to be a reduction in oxygen on the hemoglobin. So even though we're inhaling and exhaling, that gas exchange, and this would be the external gas exchange, or external respiration, is inefficient. So this is sensed by the kidney that oxygen levels are too low. The kidney releases REF, renal erythropoietic factor. REF and the globulins that are already present in the blood form erythropoietin, which stimulates bone marrow to produce red blood cells, erythropoiesis. Negative feedback is this increase of oxygen on our hemoglobin. So we get more red blood cells, we get more oxygen in our blood because we have more spots for more hemoglobins to bind oxygens to. So there's our negative feedback. Once our oxygen levels are back up, the kidney senses this, it stops producing REF, and we stop producing the extra red blood cells. It's also a homeostatic effect in maintaining body temperature. Our circulatory system maintains body temperature by using vasodilation and vasoconstriction. So heat is produced by cellular respiration when we metabolize sugar and absorb it by the blood, and it's absorbed by the blood. That heat is absorbed by the blood. Vasodilation allows more blood to travel close to the surface of the skin, whereas vasoconstriction prevents blood from reaching the surface of the skin to conserve heat. So we have two pictures over here on the right. One is of vasodilation. When blood enters these capillary beds close to the skin, we release heat, and this is going to happen on a hot day. If we're in some place that's hot, or if we're exercising, we're producing more heat, or we're storing more heat, so more of those blood vessels, more of those capillary beds are going to open up close to the surface of the skin. On colder days, or when we're not doing as much physical activity, these precapillary sphincters are going to close, reducing the blood flow to that capillary bed. Notice it's not closed off entirely, but it's definitely a lot smaller when you compare it to the picture in picture A. The other neat thing is that with the design of our circulatory system, we generally have one deep artery running through our limbs, and we end up with actually two veins that return the blood back to our heart. There's a deep vein and a surface vein. What this does with the deep vein is it maintains a constant body temperature. As heat is radiated from this limb, some of that heat ends up in the deep vein. That heat can then be returned to the body. So it's a conservation method. If all the heat made it out to this surface vein, then the heat would be lost and we'd feel cooler. So it's called a countercurrent heat exchange mechanism between blood vessels in the arm, and we have them in the legs as well. The deep vein and artery are adjacent to one another, so heat is exchanged from one to the other. As a result, arterial blood is cooled as it nears the hand, and the venous blood is warmed as it returns to the body core. So we're conserving heat. In high temperature conditions, more blood is going to return through the surface veins. So if it's hot outside, the heat's going to travel down into the capillary bed. More of that blood is going to travel through the surface vein, thereby letting us release that heat. So from this lecture, you should have the idea that blood is composed of three types of cells: erythrocytes, leukocytes, and platelets, and blood plasma. Erythrocytes are involved in the transport of, and nutrients is an okay word, but we like O2 better. Erythrocytes transport oxygen. The leukocytes are involved in immunity, and platelets initiate blood clotting. Plasma is the fluid medium for the transport of blood cells, nutrients, waste, and a variety of blood proteins with specific functions, such as fibrinogen. And the circulatory system is involved in many homeostatic processes, including blood clotting, erythropoiesis, and thermoregulation. It also transports chemical messengers for other homeostatic responses. So the circulatory system plays a large role in homeostasis.